Good morning, children. After a short break, we are commencing our classes again. Children, in our last session, we have discussed about various methods of contraception and various sexually transmitted disease. All we have discussed, isn't it? And I have given an introduction about infertility also, right? And what is infertility? It is a two-mark question. Inability to conceive or produce children even after unprotected sexual cohabitation is called infertility. That means when couples have unprotected sexual contact, though they are doing unprotected sexual contact, they are not getting conception, and this condition is called infertility. That is the inability of a man to produce sufficient numbers or quality of sperm to impregnate a woman, or inability of a woman to become pregnant or maintain a pregnancy. That means the defect may be in male or maybe in the female. So it is on both sides we can expect. We cannot blame not only the male, not only the female. Okay, the causes for infertility are tumors formed in the pituitary or reproductive organs, inherited mutation of genes responsible for the biosynthesis of sex hormones, malformation of the cervix or fallopian tubes, and inadequate nutrition before adulthood. So these are the various reasons for infertility. Long-term stress damages many aspects of health, especially the menstrual cycle. The ingestion of toxins, heavy metal cadmium, heavy use of alcohol, tobacco and marijuana, injuries to the gonads and aging also cause infertility. So in this paragraph, it's fully described about the various reasons of infertility. So you can have it as a 3-mark question, children. Please bracket it. Then what are the other causes of infertility? So in the above said paragraph, we have read out many reasons for what infertility, and other than this, some other reasons also there to cause infertility. Let's see about it one by one. Pelvic inflammatory diseases, that is PID, pelvic inflammatory disease, uterine fibroids and endometriosis are the most common causes of infertility in women. So this is what is some causes of infertility. What are the some causes? Pelvic inflammatory disease. It is a type of disease. Inflammation it causes in the pelvic region, and uterine fibroids, that is a tumor like structure, it is present inside the uterus, and endometriosis are the most common causes of infertility in women. Then low body fat or anorexia in women, that is psychiatric eating disorder characterized by the fear of gaining weight. Okay, so usually women are getting afraid when they are increasing their weight, isn't it? So to avoid that, the women are avoiding to have a food. Okay, if we take a food, more food, we are gaining weight still more அப்படிங்கிற ஒரு பயத்திலேயே சாப்பிடாம இருக்கிறது. Then undescended testes and swollen veins in scrotum. So in the scrotum, testes are present inside the scrotum, that is outside the abdominal cavity. So the testis is undescended, not in the original position of the scrotum, it is undescended, and swollen veins. These are the reasons for infertility. It is also 5-mark questions. Then tight clothing in man may raise the temperature in scrotum and affect the sperm production. Underdeveloped ovaries or testes, and female may all develop antibodies against her partner's sperms. Males may develop an auto response to their own sperms. So these are the various reasons discussed for infertility. It is a 5-mark question, children. Last year question also, please bracket it. All women are born with ovaries, but some do not have functional uterus. This condition is called what? Mayer-Rokitansky syndrome. So it is a 2-mark question. That means the women, they are having ovaries. It is a functional only, but they do not have a functional uterus. They have ovaries but not a functional uterus, and this condition is called Mayer-Rokitansky syndrome. Then next we are going to see assisted reproductive technology. What is mean by assisted reproductive technology? It is nothing but the various technology which help to have a child in an infertility couples. Okay, that these various techniques are together called assisted reproductive technology. The various technologies are we are discussing one by one in the upcoming paragraph. A collection of procedures which include the handling of gametes or embryos outside the body to achieve pregnancy is known as assisted reproductive technology. 2 mark question. What is assisted reproductive technology, or discuss about the assisted reproductive technology? Okay, it is a 3 mark as well as 2 mark question. Simple in assisted reproductive technology definition: a collection of procedures which include the handling of gametes or embryo outside the body to achieve pregnancy is known as assisted reproductive technology. Next one, list some of the assisted reproductive technology which are in practice. Okay, like this you are having some other 2 mark question also. It increase the chance of pregnancy in infertile couples, or it includes intrauterine insemination, in vitro fertilization, embryo transfer, zygote intrafallopian transfer, gamete intrafallopian transfer, intracytoplasmic sperm injection, pre-implantation genetic diagnosis, oocyte and sperm donation and surrogacy. So these are the various technologies coming under the assisted reproductive technology. Okay, so please by heart it, children, and sometimes you will have an explanation for abbreviation of ZIFT. What is ZIFT? What is gift? And define ZIFT, define gift. Like that you will have a question. So please sure of this expansion also. Intrauterine insemination is the first one we are going to discuss under this art. Intrauterine insemination. So in the topic itself we can understand the concept. Intra means what? Within. Intrauterine means what? With uterine means here uterus. So that means within the uterus. Insemination means what? Keeping the semen. Keeping the semen. That means inside the uterus, the artificially the semens are introduced. That is called what? Intrauterine insemination. Okay, this is a procedure to treat infertile men with low sperm count. Please note this sentence, children. For whom we are doing this intrauterine insemination technology? The male, those who are having low sperm count. For them, we are using intrauterine insemination. The semen is collected either from the husband or from a healthy donor and is introduced into the uterus through the vagina by a catheter after stimulating the ovaries to produce more ova. See, before we are introducing the sperm into the uterus, we should stimulate the ovaries to produce more ova by a hormonal injection. Then only fertilization will be possible. Okay, once the ova is produced, that is ruptured and released from the ovaries and enter into the fallopian tube, at the same time we have to introduce the semen into the uterus through the vagina by a catheter. Okay, then only the sperm and ovum can meet in the fallopian tube naturally and fertilization taken place. So this treatment is given for infertile man or the man with low sperm count. Then the sperm swim towards the fallopian tube to fertilize the egg, resulting in normal pregnancy. Right. Next, in vitro fertilization or test tube baby. Usually in vitro means what? Fertilization outside the body is called in vitro fertilization. What is in vivo fertilization? Fertilization inside the body of female is called in vivo fertilization. Here in vitro fertilization, that is fertilization taken place outside the body in the laboratory in under controlled conditions. Okay, that is called what? In vitro fertilization. It is otherwise called as what? Test tube baby. In this technique, sperms and eggs are allowed to unite outside the body in a laboratory. One or more fertilized egg may be transferred into the woman's uterus, where they may implant in the uterine lining and develop. Excess embryos may be cryopreserved for future use. Initially, IVF, that is in vitro fertilization, was used to treat women with blocked, damaged, or absent fallopian tubes. Today, IVF is used to treat many causes of infertility. The basic steps in an IVF treatment cycle are ovarian stimulation, egg retrieval, fertilization, embryo culture, and embryo transfer. This is a 2-mark question. Write the various steps involved in the treatment of IVF. First, ovarian stimulation. Why to stimulate the ovary? To produce more egg. For that, they are stimulating the ovary. So, once you are stimulating the ovary by a hormonal injection, the ovaries produce many eggs. Usually, in a cycle, the ovary produces only one egg. Instead, by stimulation of hormonal injection, the ovary produce more than one egg. After that, we are collecting the eggs. That is called what? Egg retrieval. After collecting the egg in the outside the body in the laboratory under controlled condition, fertilization taken place. Once the fertilization taken place, embryos are formed. Now we are doing the culture of embryo in the laboratory. After the embryo getting matured, not matured, 8-cell stage, and we are transfer the embryo into the uterus. In the uterus, the embryo implant and started to develop as a child. Then egg retrieval is done by minor surgery under general anesthesia using ultrasound guide after 34 to 37 hours of HCG, that is human chorionic gonadotropin. That means once a hormone, human gonadotropin hormone is injected into the female body, after the injection 34 to 37 hours, a minor surgery will be taken place in the female body under general anesthesia, and the eggs are removed. Okay, the eggs are prepared and stripped from the surrounding cells. At the same time, sperm preparation is done using a special media. When one side is egg preparation has taken place with the minor surgery, another one side sperm preparation also going on. After preparing the sperms, the eggs are brought together. So, after preparing the sperm, the eggs are brought with the sperm. 10,000 to 1 lakh motile sperm are needed for each egg. Then the zygote is allowed to divide to form 8-celled blastomere and then transferred into the uterus for a successful pregnancy. Then the transfer of an embryo with more than 8 blastomere stage into uterus is called embryo transfer technique. Okay. See children, when you transfer, usually the procedure, as per the procedure, the egg should be transferred into the fertilized egg should be transferred into the uterus in 8-cell stage. When you transfer the embryo after 8-cell stage, then it is called what? Embryo transfer technique. See here it is given as a cryopreservation. What is cryopreservation? Please do you know? You complete it children. Next I am going to say about ZIFT. What is ZIFT? Z for zygote, I for intra, F for fallopian, T for transfer. That is called what? Zygote intrafallopian transfer. And from this also you can understand what is mean by ZIFT. That is zygote intrafallopian. Intrafallopian means what? Within the fallopian tube or inside the fallopian tube transfer. What they are transferring into the fallopian tube? They are transferring zygote into the fallopian tube. Right children? As in IVF, the zygote up to 8 blastomere stage is transferred to the fallopian tube by laparoscopy. The zygote continues its natural division and migrate towards the uterus where it gets implanted. See, usually in natural fertilization, the fertilization has taken place in the fallopian tube, right? In the fallopian tube, fertilization has taken place and the zygote undergo mitotic cell division, 1 cell into 2, 2 to 4, 4 to 8, like that it's going on inside the fallopian tube. And it rotates coming on the way to the fallopian tube and reach the uterus, isn't it? The same way, we are introducing the 8-celled stage of zygote into the fallopian tube. From the fallopian tube, this zygote it's coming through the fallopian tube and reach the uterus. There it's get implanting as like natural path. Okay, that is called what? ZIFT. Next one is IUT, intrauterine transfer. So that is what transfer of embryo here. There intrauterine insemination, first topic we have discussed, isn't it, children? Insemination, we are depositing semen inside the uterus. That is called intrauterine insemination. Here intrauterine transfer. What is mean by intrauterine transfer? They are transferring embryo into the uterus. They are transferring embryo into the uterus. That is called what? Intrauterine transfer. Embryo with more than 8 blastomere is inserted into the uterus to complete its further development is called intrauterine transfer. Embryo with more than next one is gamete intrafallopian transfer, that is gift. Transfer of an ovum collected from a donor into the fallopian tube. Here in gamete intrafallopian tube, okay. In this, the egg are collected from the ovaries and placed with the sperm in one of the fallopian tubes. The zygote travels towards the uterus and gets implanted in the inner lining of the uterus. Okay, here gametes are transferred into the fallopian tube. Gametes are transferred, both the gametes. Transfer of an ovum collected from a donor. So, some females cannot produce a capable egg. So that egg, her egg cannot have the ability to form a zygote. So in that case, they will go for donor egg. So the donor means what? Some X or Y female can donate her egg to that inability female. So they have collect that donor egg and introduced into the fallopian tube. And same way the semens also collected from outside and introduced into the fallopian tube. There the egg and sperms are getting fertilized and form zygote, and the zygote pass through the fallopian tube and attached in the uterus, and this is called what? Gamete intrafallopian tube transfer. Transfer. Next, intracytoplasmic sperm injection. You see, intracytoplasmic, that is within the cytoplasmic sperm injection. Sperm வந்துட்டு எக்கோட சைட்டோபிளாஸ்த்துக்கு உள்ளே இன்ஜெக்ட் பண்ணுறது. இது யாருக்கு நடக்கும் அப்படின்னு பார்த்துட்டிங்கன்னா, ஸ்போமோட கவுண்ட் ரொம்ப கம்மியாக இருக்கிறவங்களுக்கு, ஆக்டிவிட்டி ரொம்ப கம்மியாக இருக்கிறவங்களுக்கு இந்த மாதிரி மெத்தடை ஃபாலோ பண்ணுவாங்க. ஸோ லெட்ஸ் சி. In this method, only one sperm is injected into the focal point of the egg to fertilize. The sperm is carefully injected into the cytoplasm of the egg. Fertilization occurs in 75 to 85% of eggs injected with the sperm. Then the zygote is allowed to divide to form an 8-cell blastomere and then transferred into the uterus to develop a productive pregnancy. So this intracytoplasmic sperm injection also taken place in the laboratory, not in the female body. Okay. Next is surrogacy. What is mean by surrogacy? Surrogacy is a method of assisted reproduction or agreement whereby a woman agrees to carry a pregnancy for another person. So the vadagathai, you can heard this word previously, I think so. வாடகை தாய், அதாவது ஒருத்தவங்களோட குழந்தைங்கள அது the X or Y female uterus is maybe very weak. So in that case he cannot carry her own fetus. So in that case they will go for another woman to carry that. So they are that process is called what? Surrogacy is a method of assisted reproduction or agreement whereby a woman agreed to carry a pregnancy for another person who will become the newborn child's parent after birth. Through in vitro fertilization, embryos are created in a lab and are transferred into the surrogate mother's uterus. Next, male infertility. A zoospermia is defined as the absence of spermatozoa in the ejaculate semen on at least 2 occasions and is observed approximately in 1% of the population. A zoospermia is defined as the absence of spermatozoa in the ejaculate semen. There is no spermatozoa even in a semen on at least 2 occasions and is observed approximately in 1% of the population. This is called what? Male infertility. Then microtesticular sperm extraction, that is called what? TESE, T-E-S-E, TESE, microtesticular sperm extraction. Microsurgical sperm retrieval from the testicle involves a small midline incision in the scrotum through which one or both testicles can be seen. Under the microscope, the seminiferous tubules are dilated and a small amount of testicular tissue in areas of active sperm production are removed and improved for sperm yield compared to the traditional biopsy techniques. Okay, what they will do here in the test is, they will do some incision in the scrotum, and then they can see a 2 or 3 seminiferous tubules. Under microscope, they have to remove some testicular tissues where the active sperm production are produced, and this sperm yield is improved when compared to the traditional biopsy technique. And next one is detection of fetal disorder. So what is fetal disorder? The disease which we can be seen in the developing fetus inside the uterus is called what? Fetal disorder. Then how to, since the uterus is inside the mother's womb, we cannot find the condition of the fetal. Then in that case, how to detect the disorder in the fetus? They are using some method to detect that. One is ultrasound scanning, another one is amniocentesis, okay, and the third one is chorionic villus sampling, then fetoscope. These are the various methods to detect the disorders in the developing fetus. Okay, let's see about ultrasound scanning. Ultrasound has no known risk other than mild discomfort due to pressure from the transducer on the abdomen or vagina. So ultrasound has no known risk. That means there is no risk at all when the procedures are going on other than mild discomfort due to pressure from the transducer on the abdomen or vagina. So no radiation is used during this procedure. Ultrasonography is usually performed in the first trimester for dating, determination of the number of fetus, and for assessment of early pregnancy complications. Okay, so the uses of ultrasound scanning is they are usually doing in the first trimester to detect the what is the dating, when the pregnancy date will be, not a pregnancy date, when the delivery date will be, then how many fetus are there, that means what, 1 or 2, sometimes 3, 4, like that. So that to detect the number of fetus inside the uterus and for assessment of early pregnancy complications. Okay, these all we can see under this. Then next one is amniocentesis. This you know very well, already I discussed with you about amniocentesis. The fetus is floated inside a fluid called the amniotic fluid. In the amniotic fluid, some cells of the fetus are present. So amniocentesis is a technique when they suck the amniotic fluid through a injection, and in that fluid we can find some cells of the fetus. That with that cells we are doing karyotyping, and if we see any chromosomal abnormalities or any deletion or additions or any gene malfunctioning, everything we can find. So with that we can find out the genetic disorders of that fetus. In that case we can give a counseling to the couples to abort or terminate the pregnancy, and this is called amniocentesis. Amniocentesis mostly used for this purpose only, but nowadays the couples, they are taking it as advantage to find the sex of the fetus to abort the female infant. Okay, so government, that's what banned this technique. Amniocentesis involves taking a small sample of the amniotic fluid that surrounds the fetus to diagnose for chromosomal abnormalities. Amniocentesis is generally performed in a pregnant woman between the 15th and 20th week of pregnancy by inserting a long thin needle through the abdomen into the amniotic sac to withdraw a small sample of amniotic fluid. The amniotic fluid contains cells shed from the fetus. Then next one is chorionic villus sampling, that is called CVS, as a prenatal test that involves taking a sample of the placental tissues to test for chromosomal abnormalities. Next one is fetoscope, is used to monitor the fetal heart rate and other functions during the late pregnancy and labor. So when a female get a pregnancy in his later age, then there will be some malfunctioning we can find in the fetus. In that case, we are using fetoscope to find the heartbeat in between 120 to 160 beats per minute. An abnormal fetal heart rate or pattern may mean that the fetus is not getting enough oxygen and it indicate other problem. So if abnormal in the heartbeat of fetus, what happened there, we can understand that the fetus cannot get enough oxygen. If it is not getting enough oxygen, it indicate the other problems also. This is called what? Fetoscope. Okay children, this lesson we have completed with this, I think. Yes, a handheld Doppler device is often used during prenatal visit to count the fetal heartbeat. Then during labor, continuous electronic fetal monitoring is often used. Vitamin E is known as anti-sterility vitamin as it helps in the normal functioning of reproductive structure. Sex hormones were discovered by Adolf Butenandt. 11th July is observed as World Population Day. 1st December is World AIDS Day. NACO, National AIDS Control Organization, established in 1992. Syphilis and gonorrhea are commonly called international disease. Underline it. So these are some 1 mark as well as some 2 mark question you will have, children. Okay, hope we completed this lesson with this, and you are clear with that, I think so. And regarding homework, today you want to complete the various technique which are involved in art. What is art? Explain the various technique involved under the art. Okay, this is the only homework, only question you have as a homework. Complete it, children. Bye.